It's now time to introduce the idea of the cell cycle, where cells have a moment of birth, a period of growth, and then they reproduce. And we're going to divide the cell cycle into two major sections, colored here red, and then the blue wedge up here. First, we'll see what happens in the red phase of the cell cycle. We're going to call that interphase. And we can think of what the cell is doing here is growth and metabolism. So to grow and to just live, cells are going to be needing to make messenger RNA, which are temporary copies, to build proteins. They're going to build the proteins at the ribosomes. They're going to be processing energy, getting energy somehow for their environment, etc. Here we see examples in the case of amoeba. On the left we have protein construction. So here in the amoeba, if we look in the nucleus of the cell, we see 3 genes are currently active. So we see messenger RNA being built from one of the strands in each of the genes here. The messenger RNAs will leave the nucleus out to where the ribosomes are located. And at the ribosomes will sort of move down the messenger RNA 3 letters at a time, attaching amino acids in the sequence determined by the sequence of messenger RNA triplets here. So we see the 3 different messenger RNAs are involved in building 3 different proteins that happen to be involved in pseudobot control. Well, cells during this growth phase are also processing energy. They're capturing food, digesting food, absorbing the nutrients to keep the cell alive. So during this red period, the cell is doing its basic life functions, getting energy, building proteins, and staying alive. Now during this phase, the cell is using the information stored in chromosomes. So it's accessing the information in genes. And we say accessing in the sense that the cell will actually have to separate the DNA strands, build messenger RNA, and then have the ribosomes make the protein itself. So in order to get at the information stored in some section of DNA, it has to be unwound, as it were. Recall those proteins called histones, our sort of packaging proteins. Well, in order to make a protein, the cell will have to unwind the gene so that it is accessible to the proteins that do the whole business of making messenger RNA. In this picture then, again, we did note that the DNA was accessible. Here we see some DNA that's wrapped up around the histone protein. Those genes are currently not accessible. But the 3 genes for making these 3 proteins are accessible. Here we see paramecium, the nucleus of the paramecium. Here's a bit of DNA. Maybe there are 2 genes here, 2 messenger RNAs. They leave the nucleus, hook up to ribosomes, and 2 different kinds of proteins are being made. Here's a digestive protein here that will do its job inside a food vacuole, and here's a transport protein here that will transport nutrients out of the food vacuole back into the cytoplasm. Here I just wanted to point out that what happens to these messenger RNAs is that they will degrade. Over here we see a little picture of the messenger RNA breaking apart. And this is what makes the messenger RNAs a temporary copy of the recipe for a protein. Here's this messenger RNA is degrading, being broken up. So DNA is the permanent recipes. Messenger RNAs are temporary copies of the recipes to build proteins. Again, let's review what it means for DNA to be uncoiled. It is detached. It is unwound and detached from those packaging proteins. If the cell is going to prepare for cell division and duplicate its DNA, the DNA must also be unwound from the packaging proteins. So the DNA has to be accessible both to make a protein and for DNA to be duplicated. So during this red phase then, in addition to just doing normal life activities for a cell, the cell must duplicate its DNA. And chromosomes, remember, are just long stretches of DNA that have many genes. So chromosome duplication amounts to DNA duplication. And we've learned how that process happens. The 2 strands separate, the extra letters match up according to the pairing rules, and then DNA polymerase comes along and stitches them together, chemically bonds them together. This duplication happens during this red phase, during interphase of the cell cycle. So now the cell will be prepared to deliver chromosomes to the daughter cells. Now, in the picture on the left, we see a bit of DNA that has been duplicated. As it turns out, when the cell duplicates its DNA, it doesn't fully finish the process, and this means that the chromosomes, the duplicated chromosomes, can still be attached at a small region here. And that will be important for the cell to just kind of keep track of its duplicated chromosomes. So the identical duplicates are still going to be stuck together at this central location here. Then what happens is that the DNA is going to coil up around all those packaging proteins. And so here's just a cartoon version of what you have then, a duplicated chromosome that's all been coiled up. So this chromosome here is one chromosome, and here is the duplicate, but they're still attached in the middle. What the cell will do then is deliver one of these chromosomes to one daughter cell and the other chromosome to the other daughter cell. In our picture then, here we see on the left, this is what that duplicated chromosome would look like. It is super coiled up. So we have one chromosome here and then one chromosome here, but they're still attached at the center, and this allows the cell to sort of keep track of its duplicated chromosomes. So now we're up here at this stage here, we're preparing for the blue part of the cell cycle, and that's called mitosis. We can think of it as the nuclear division. So the contents of the nucleus now are going to be delivered to each daughter cell. And what is in the nucleus now are duplicated chromosomes. So mitosis is this process where duplicated chromosomes are going to be delivered to the daughter cells. On the left here, we see our cell cycle. So again, the red section was interphase. DNA duplication occurred during this stage here, and now the cell is prepared to go through mitosis, where the cell is going to be moving the duplicated chromosomes into the daughter cells. The first thing that happens is the nuclear membrane will break down, and then a set of proteins, in fact microtubules, on both ends of the cell are going to search out the duplicated chromosomes and make contact and line them up. And then the duplicated chromosomes will finally detach and move to opposite sides of the cell. Now we're looking at it from the whole cell point of view. Now the cell membrane pinches in, two cells are going to form, and a nuclear membrane forms around each set of chromosomes. So this is mitosis. It's the process whereby the cell is going to deliver a set of chromosomes to each daughter cell. Going into the first stage of mitosis, though, the chromosomes were duplicated during interphase. Here we see an actual picture from a plant cell. So here we have one plant cell, and its chromosomes had been duplicated, and we have caught the cell in the act of separating the identical sets of chromosomes. If we could see all the proteins involved, we would see these microtubules that are attached to the chromosomes, and the chromosomes are kind of being moved to opposite sides of the cell, so they kind of drag behind the force of the pulling here. There's some evidence that actually they're driven along these microtubules, but that's for another time. But here we see the sets of chromosomes are being delivered to the daughter cells, and then the cell's going to divide right in the middle here, and then make a nuclear membrane around this set of chromosomes and a nuclear membrane around this set of chromosomes, and you'll have two new cells. So very importantly, we want to think of mitosis then, where the cell is moving its duplicated DNA to make two new nuclei in two daughter cells. Whereas during interphase, the cell is using its DNA to make proteins. So that's our familiar picture up here. The cell's using the DNA information to make proteins during interphase. The cell is moving duplicated chromosomes during mitosis. So that's our summary then of the cell cycle. During interphase, the DNA must be uncoiled, so it's accessible to either make proteins or to duplicate the DNA itself. During mitosis, the DNA gets all coiled up, so the cell can keep track of those duplicated chromosomes and deliver a full set of chromosomes to each daughter cell.